So we had a tremendous amount of questions that were gathered from the community over the last 2 weeks or so, and we have collated them with 2 pages in which we have to accomplish in 15 minutes to all of you. So no, I won't do that, I promise. So we're going to do a lightning round of questions, and I'm going to start with the most commonly asked question, and we don't need like a diplomatic answer, we just need a straight answer, because then we can get through to the next question. So the first question is going to go to all of the programs that are currently in the clinic, which is going to be Ionis, Ultragenyx, and Neuren. Is going to be, the community would like to know, now that you are sharing that you're in your phase 1/2 and some of you are ending your phase 1/2, can you give an expected window of when a phase 3 might start? Would you be able to answer that, Kemi? We'll start with Kemi. Actually, I'll give you another mic.
So I can start, Allison. So yeah, that's a common question that we get, and I think the difficulty in giving a definitive answer is that if you miss that time point, then you get held to it, and then you disappoint people. So, I mean, we're definitely going to phase 3. That's the intent. I think best case scenario is second half of next year, 2024. Hopefully that helps.
And everyone acknowledge, best case scenario means lots of crazy things happen in drug development, so nobody can be mad if that doesn't actually happen, because chances are that won't actually happen. But that's best case scenario. We have to be realistic as well as a community, so thank you for giving an answer. But the fact that they're committed to a phase 3, I think we should be really excited. Becky?
Is it me?
That's
Is
you.
on? Okay.
It's on.
So the shorter answer is it's too soon to tell. I mean, that's just the straight truth of it. And we're collecting the data. We're encouraged by this early data. We're certainly planning for success. So if everything keeps moving in the right direction, we hope to have, you know, the data readouts mid next year. Hopefully we'll have an update then.
Sounds good. So we'll have an update hopefully at the next conference or at the ASF conference in the summer. Great. And Nancy, are you online?
Likewise, our phase 2 is expected to read out next year. So after we have the full results from the phase 2 study, then we'll be able to give a more reasonable update about what will be happening next for the studies to follow.
Great, thank you.
Okay, so my question is for Dr. Wilson. So theoretically, in the hopes that the gene therapy moves on through approval, could patients also use an ASO on top of that gene therapy?
You know, there's precedent for that, which in SMA, which is Zolgensma and Spinraza. And there are situations where patients who receive Zolgensma didn't respond and then are provided with Spinraza. I don't think there's any reason from a biological standpoint they wouldn't. The problem I think you run into is payers. They don't like to pay twice.
Yeah. That's very true.
And the data will show whether it's needed, hopefully. Okay, this question is for Stephanie. You're there, I see you. Of course, it's a similar question, which I think I know the answer, but in respect to the question from the community, if all goes well, what are your anticipations on a timeline that you would see in which you would be considering starting a phase 1?
Good question.
Too early to tell. I know that's the answer.
It is too early to tell. What I can tell you is that we are currently in non-human primate studies with these updated optimized candidates, and that data will be reading out early next year. And based on that information, we'll know whether we can proceed directly to IND-enabling studies or whether more optimization will be needed.
Great. Thank you.
Great. I want to make sure we get questions from the audience. Do we have
Does anyone in
our
the audience
first
want to raise their
question?
hand?
We've
Over
got
here.
one over there.
Oh, right there.
And then we got one over here. Okay.
Yes.
Oh, go ahead.
Anthony from Fast France.
Hi.
Yes. Yes. The first domain that the family would like to improve is the communication. It's the same in France and in USA. And it's the same in the pediatric population and adult population. FAST invest in the ORCA scale And yesterday we had a presentation from Dr. Reeves to give interpretation about the scale and the improvement. It's very concrete for my family. I would like to know why in the current clinical trial we don't have this outcome measure in a secondary outcome measure, and when we have this, scale, we have some result with the Bayley 4. Is there a problem with this scale because it's a caregiver scale, or yes, or another issue? Thanks.
There was a big echo, and I couldn't actually understand what scale you asked for. Megan or Bianca, can you? The ORCA. Okay, I'm sorry, I couldn't understand it. Are you asking that question directly to someone on the stage, or to me? Okay. Those in clinical trials, you're wondering what are the outcomes of people using the ORCA?
What are they saying?
What are they
saying? Is the question if we're using the ORCA? Is that it?
The question is, what are the results of the ORCA, or if they're using the ORCA? Both. Both.
So I can say we're using the ORCA. We don't have results on it yet. I mean, we're going through that data collection now, so we'll be able to report on that at the completion of the part 1, but we are using it.
Kenny? I think it was to everyone in the clinic.
This is Nancy. I'm on the line. We're also have the ORCA in our study, but we're still completing the study, so we don't have results yet, but we're using it.
Yeah, and the same for Ultragenyx. So we do have the ORCA in our trial, but we don't have sufficient data right now to be able to draw any specific conclusions, but more to come.
And I think from a reality perspective, the community needs to know that it takes time to crunch all of this data. It's quite tremendous. And so if people are willing to give small data cuts, it doesn't mean they have all of it. And so we have to be patient as things roll out at different times, because the way you analyze that data can be tremendous and take a real lot of time. So if the trial's not done, they're not going to have data to share. If the trial is getting cut at different time points, there's going to be different time points of people in the trial at that point for data to share. So it's very hard to expect everything. There was a hand up here.
We had one here. There's one back there.
Okay.
Okay, this is for Elizabeth. In the survey, have you considered to include questions related to understand the mental health for the caregivers and or the parents when they receive the news that their child are affected, or then when they are, you know, in the middle or struggle with taking care of the kids? Thank you.
Yep. So there's a couple of ways that we capture mental health in the survey. I mean, one thing that is important to remember in this survey is that we're looking for things we can quantify. So we do ask questions about your use of mental health treatments and money that you spend on those. We also asked, we were looking back over a year, so that doesn't necessarily capture everyone's loved one's diagnosis. The other thing that we capture is a measure called quality of life, which is very commonly used in health economics. A component of that relates to mental health, and for those of you who took the survey, you may remember answering questions related to your mental health that fit into those quality of life instruments. And that's something that using health economic principles, we can put sort of a quantified number on. So yes, there are components of mental health. I'm sure there's a lot more work to be done beyond what was in our survey.
Great, thank you. There's a question here.
Okay, hi. So I'm talking specifically about mutation patients and GTP 220 or therapies like them, right? So wouldn't some of the clean gene that's being expressed be picked up by the receptors along with what I understand to be a glitchy gene that mutation kids are expressing, right? And theoretically, if that was picked up by the receptors, wouldn't that support a gain of function? And then my real question is if that's true, right, or if that's theoretically true, then why aren't our kids in trials?
So you're asking if there's a gain of function. So not all kids with mutations have have maternal gene that's being expressed, but if they do, so some of the missense mutations, and those are being expressed, then will GTP220 benefit those kids?
Exactly. And then why wouldn't you then put them in the trials, right?
Do you want to answer that, or do you want me to try to answer it?
Yeah,
Actually,
it's
Liz,
a
you
really
can answer that.
it's a really good question. Every loss of function disease that we've been involved with raises the possibility that a missense mutation actually creates a protein that, if complexes with a normal protein, can interfere with its activity. That's generally the way in which a mutation could negatively impact on the function of a gene replacement. A few times we've tried to test that and never been able to confirm that, and so it hasn't seemed to be a problem in any of the gene replacement studies. You can study it in vitro, but we're still in discussions about eligibility criteria for the gene therapy program, and I think that's an important point that we ought to consider.
And Liz has experience with this.
I think there might be some clues, because 2 of the ASO studies are enrolling mutation patients, and so there might eventually be some clues about that. We've thought about that a lot, you know, in terms of enrolling missense patients into those trials.
Great. Did that answer your question?
Hi, my name is Lauren, and I'm a UPD mom. So this question is for Becky and Kimmy. Becky, I know that you said that you didn't know when UPD and ICD would be included in an Ionis trial, but can you give us a date? Assuming everything goes well and perfectly, can you give us a date by which you would give us a date? We gotta have something to cling to.
So you want a date for a date? So our data readout from, as I mentioned, from the Part 1 MAD will be mid-next year. I think that's going to tell us a lot about next steps and how we can move our way into the broader population. We also have the Part 2 long-term extension going on, and I don't want to discount the richness of that data and how informative it's going to be. It's going to tell us about longer-term dosing, clinical benefits, that sort of thing. So we're going to get to the MAD1 data cut next year, and we'll be able to mine that data and understand it better. That may tell us something about broader populations we can go into. It may tell us about a phase 3, but it might not tell us everything we need to know. We may need some of that longer-term data to help make really good, informed, research-based, data-driven decisions about that. It's just too soon to tell right now. They're coming. I mean, this ASO approach theoretically can treat all the mutations. There shouldn't be a reason why it can't. I think it may just have to be approached in the clinic differently, whether with a dose titration or a certain dose level or a certain dose interval. And so the information we gather right now from the kids who have one dad's copy, that'll help inform on the next for kids who have 2 dads copied. We keep building on that knowledge. So we can't predict when we're going to have all the data we need to make a really thoughtful decision around that. But we're doing the studies to get there.
Thank you. And then Kimmy, assuming everything goes perfectly, which I know is a big assumption, is there a date by which Ultragenyx is aiming to include UPD and ICD in a trial?
There is. So the plan is to do that concurrently with phase 3, but it'll be sequential. So phase 3 will start before studies in the other. Phase 3 in deletion patients will begin before a study or studies in other genotypes. So there'll be some overlap, but it'll be sequential but fairly quick. So I think when we have a firm announcement about the beginning of phase 3, then we'll also have information about the other genotypes.
Great, thank you. And just for those mosaic families who I know never get talked about because there's so few of you, but many are here, The same is true for those as well, and we've had multiple discussions about that. That is the next step as well. And so when they say all genotypes, the goal is eventually that the same will apply, because most mosaicisms are ICD, and the same will apply, that it's going to be time and sequential, but we'll ensure that every genotype sees their day, for sure.
Question back there. Go ahead.
I've got a question for Roche. So last year, the conversation was around the strategy between upstream and downstream working together, so, right, ASO with Allogabat. Contract negotiations aside, just wondering, with Roche kind of retracting from the ASO side, has that strategy changed with just the focus on Allogabat?
So if I understand your question correctly, you're asking if we shifted the strategy from ASO to Allogabat. Is this the question?
Well, the strategy was for upstream and downstream to work together, hopefully, right, with whatever happens out of the trial. So I'm asking, with getting out of the ASO side, has that strategy for Roche changed to just focus on downstream? Or is it downstream and working with other ASOs, like with Ionis and Ultragenyx?
So, in fact, what the regonursing program, the data which we got from the interim analysis was not sufficient for us to move forward with a next stage based on the internal criteria. It has nothing to do with downstream or upstream or changing strategy. So every company has its own internal criteria to move from program forwards, and this program did not meet our internal criteria, so that was the decision taken to hand over the program to potential partner. Did I answer your question? Or?
Isabel, and then
yes, here.
We'll go to the back, but go ahead.
Hello, and thank you to all of you. I am Marlene Dominguez from Nicaragua, Central America. My daughter Silvana has a diagnosis of 93% of deletion type, and 7% is typical. So my question is that considered a mosaic type or a deletion type? And that leads me to my second question. If we have any possible treatment or clinical trial for my child, would she be treated as deletion type or mosaic type? Thank you.
Liz, you should probably take that.
I don't
can
know
make an
that,
assumption on this, but.
I don't know that we have specific criteria for mosaics. I mean, she certainly is the deletion type, but she is also mosaic. So she's really both of those things. And that would depend on whether there were criteria that said that we can't enroll any mosaic individuals in a trial. And those criteria for phase 3 haven't been defined yet. So I think we'd have to wait and see. The concern would be that in the mosaic cells that don't have the deletion, that you could be activating the paternal gene, and then you would have double the amount of UBE3A in those cells. But if it's only 7%, it probably wouldn't be a big issue. So I think those are criteria that we're probably going to have to deal with in phase 3 design.
Yep, question all the way in the back.
Yeah, hi there. Thank you for your transparency with these trials. I have a question. In the results that you report, the improvement in the outcome measures, in the mean or median or however you're reporting that, my question is really about the outliers beyond the standard deviations of what you may have found. Again, I know it's preliminary in the patients that have better responses and the patients that you're not seeing responses. Do Do you get a sense that the more impacted individuals are having more of a response? Do you get a sense that the older patients are not having as good a response, or it's even across the board? Some are mixed genotypes, and I know some of you have mentioned this, but I'm just wondering, in your overall review, have you appreciated anything about the outliers?
We'll start with Kemi. Or maybe Liz. So you could just be non-company specific.
Non-company specific. You know, every drug on the planet has variation in response. And I think it would be surprising if we didn't actually see that. And it could be related to age, to severity, to some people may even have like a mutation in the AS gene, the antisense gene that may preclude the binding of, you know, one particular ASO or another. And those are things that we just don't have enough patients yet to work out yet. I mean, that's a very good question for the future as we go forward and treat hundreds of patients.
And we've talked about this with Yang Wei, about as patients enroll in these trials, and we're assuming that the antisense transcript is intact and there's no SNPs or polymorphisms in the antisense transcript, but are we saving their blood to fully genotype the antisense to understand if the non-responders may actually have a polymorphism in the sequence of the drug, in which one ASO would be better for that patient because they're both in different sequences than another ASO. So something to certainly consider as a community in advocating for saving the blood and maybe getting full genome sequencing on these patients, as we can look back at responders and non-responders to try to understand that.
And so you're right. Eventually, after we have more patients to study, understanding those outliers is going to help us with the biology for sure.
Yeah.
Isabel.
Hi, my question is for Ultragenyx. Knowing that the first original patients were on a much higher dose of the GTX-102 and had the most significant improvements, and now the cohort seems to be blocked at 14 mg. Are you looking for to increase that 14 mg limit in the future, furthermore in the trial?
So, if I could just rephrase your question, you're asking if we plan to go above 14 milligrams, or you're asking if the improvements that the initial patients in cohorts 1 to 3 displayed are seen in the lower doses?
I'm asking if you're looking towards increasing the 14 mg limit.
So, I mean, we're at the tail end of phase 2, and you know, in phase 1 and 2, you evaluate safety and you optimize your doses. So we're not done. So I can't really categorically tell you what the doses are going to be in phase 3, but I can say that we are seeing equal efficacy in the high doses that the patients received very, very early on in 2020 compared to the therapeutic doses now.
And we have a last question over here, and then we're going to wrap it up.
Hi. I would guess that most of the children here are on medicine, or possibly a lot of medicine to treat seizures, to treat behaviors, to treat constipation, to treat everything. My adult son takes a lot of medicines. Would that preclude him from being in a clinical trial? How would you handle the doses that they're getting now versus during the trial and after the trial? Is the goal, my goal would be for him to be off medicine completely. But when would that happen?
I'm going to give that to you, Liv. I think for you.
So in the studies to date, medicines are not really prohibited. It's more the medicines have to be stable for a certain period of time before the patient starts the study, just because we don't want people who are having an acute seizure exacerbation to come into a study. So patients have to be stable, but there's no limit to what you can be on for constipation or for seizures or whatever. We'd love to be able to wean people's seizure medicines, and that's kind of a clinical decision. And there are some people who have weaned seizure medications after going into these trials. And hopefully that's an outcome that we can measure at some point once we have more information. But the trials to date have not really restricted clinical management of problems that go on with the patient during the study, and you know, adjustments of it, we aren't told we can't adjust doses over the long run. Because when you think about it, these are studies that are going to go on for years. And keeping an Angelman patient's medications exactly the same for years would be a daunting task, given all of the various ups and downs and things. So I don't think any of that will be a limitation.
Great. Okay, we're going to wrap it up.
Great. Thank you so much to our speakers, to all our presenters, and to everyone who attended. You know, you'll be able to talk in the hallways, ask your questions, gentle stalking, nothing serious. Don't follow them to their hotel rooms. But thank you. We can't thank you enough. You know, this is where hope lives in the people that we heard from yesterday in the presentations and the speakers from today. We're going to continue to try to convene these meetings and webinars and in person so you can ask your questions and that we can get answers. So thank you all. Thank you for being so welcoming to me for my first summit. Looking forward to my first gala. I want to thank the FAST staff for an amazing job, and Laura, who has been keeping everything organized. You are incredible. To Open Health, to Encore, and to the hotel. But thank you so much. We really look forward to seeing many of you tonight. And for those who we don't see, have a safe trip home, okay? Thank you so much.